Earlier stages of development, at the earlier stages of human development, um, the primordial germ cells divide by mitosis, resulting in proliferation and increase in the number of the germ cells. And you know that mitosis contains many stages that we may classify them as the following. Probably this is a summary of the stages, which are prophase. In the prophase you can see the chromatin of the DNA start coiling to form the visible chromosomes. The centriole is starting to be splitting and forming the microtubules of the mitotic spindles. After DNA duplication in the prophase, the double structured chromosomes are arranged in the middle of the cell at the metaphase stage. Each piece of the double structured chromosome is called the chromatid. These double structured chromosomes arranged in the equator of the cell, connected by microtubules to the centriole. Then the centromere connecting the two chromatids will be splitted, and thus the chromatid will be separated by contraction of the microtubules in anaphase, followed by beginning of telophase, after which the cell will divide into two daughter cells. The sum of each stages will be seen in meiosis, but with many differences. After that, the cells will start to proliferate. They will form two-cell stage. And as a result of division, there will be also division of the first polar body, so the polar body also will form two secondary polar bodies. The cells divided after fertilization are surrounded by zona pellucida, and each of the cells is called blastomere. And you can see this is two-cell blastomere formed of after fertilization. This is 4 blastomeres and so on. On the third day, when the number of the cell will become about 16 in number, the structure will be just like a mulberry, and it will be called as the morula. The morula will enter the uterine cavity, and inside the uterine cavity, fluid will accumulate inside the inner cells of the morula to form the so-called blastocyst. So this demonstrates the blastocyst with a cavity which is a blastocele cavity. The inner cell mass is seen around there, and the blastocyst is implanted into the uterine mucosa. This implantation occurs in the first week. In the second week, the inner cell of the blastocyst will differentiate, forming columnar epiblast and cuboidal hypoblast. The hypoblast will be near the blastocele cavity. Also, the trophoblast, the white in color, will be differentiated into two layers, starting from the embryonic size, which are cytotrophoblast, the mononucleated, and syncytial trophoblast. Also, amniotic cavity appear in the epiblast that is roofed by the amnioblast but floored by the epiblast. These changes will continue in the second month. Embryo. Here also you can see epiblast and hypoblast. Amniotic cavity roofed by amnioblast. The hypoblast will form a tissue lining the blastocele cavity, which is the yolk sac. The yolk sac will be formed in two stages, primary and secondary. The primary will be pinched off and then degenerate as a primary yolk sac cyst, or called sometimes Hesser cyst or exosomic cyst, while the secondary yolk sac will remain and continue during the later developmental stages. Also, the secondary yolk sac form a tissue to the outside, which is called extraembryonic mesodermal tissue. This tissue will be cavitated. The white color represents the cavity of the extraembryonic tissue, and this cavity will divide the extraembryonic tissue into a visceral layer near the yolk sac and amnion, and somatic layer near the cytotrophoblast. The somatic layer of extraembryonic mesoderm sometimes is called as the chorionic plate. Such cavity of the extraembryonic mesoderm is also sometimes called as the chorionic cavity. And you can see that the region connecting the embryo to the cytotrophoblast remain as an intact extraembryonic mesodermal tissue, which is not cavitated, and thus remaining connecting the embryo to the cytotrophoblast and thus forming the connecting stalk. This demonstration in the second week also shows that there are many villi formed of cytotrophoblast and surrounded by syncytium. The central core of these villi is the somatic extraembryonic mesoderm of the chorionic plate, and thus these villi are the tertiary villi. At the third week, the gastrulation process occurs by formation of the primitive streak. You know that the anterior end of the primitive streak represents a transient canal called the neuroenteric canal connecting the amnion with the yolk sac. Again, this is the amnion and this is the yolk sac. In the third week, the gastrulation in the primitive streak will transform the parts of the germ disc anterior to the streak from two layers in the streak, which are epiblast and hypoblast, into three layers, ectoderm, mesoderm, and endoderm. However, this section is a mid-sagittal section, so the layer representing the mesoderm is represented by the notochord. Later in the third week, there will be beginning of neural plate formation and heart formation, and so you can say that organogenesis begins in the anterior part of the trilaminar germ disc, while the posterior part is still showing primitive streak and showing gastrulation. Also in the third week, you can see that there is a diverticulum from the posterior wall of the yolk sac called allantois. This allantois is vestigial in human, but in animals it serves for excretory function. The allantois could be seen in the connecting stalk, and you can see that these are the mature branching villi. Each of them is formed of central stem villi and the branching terminal villi. In between the villi, this is the intervillous space. You can see that the blood vessels in the villi are connected with the blood vessels in the chorionic plate, and the blood vessels in the chorionic plates are connected with the blood vessels in the connecting stalk. Blood vessels in the connecting stalk are connected with the intraembryonic blood vessels. So this is the figure in the third week of development showing the trilaminar germ disc formation. In the fourth week, which is 1 month of age, organogenesis will proceed because organogenesis begins late in the third week and continues to the 8th week. So this is an embryo of 1 month of age. You can see the dorsal somites, which are arranged as segmental tissue blocks on the back. You can see also the pharyngeal arches as tissue blocks on the side of the neck. You can see that the amnion is enlarging surrounding the embryo. Still we have chorionic cavity, which is cavity of extraembryonic mesoderm. Still we have some parts of the yolk sac, and this is the connecting stalk. Here also you can see that some of the villi in the region which is away from the umbilical cord, starting to be degenerating to form the chorion leaf or the smooth chorion, while the villi adjacent to the attachment of the umbilical cord are growing to form the chorionic frondosome. In the second week, the embryo is more differentiated. Still, the somites are seen, still the pharyngeal are seen, but here we can see also that the limb buds are formed in the second month. The eye primordium and the ear primordium are seen on the sides of the head. Also, you can see the olfactory primordium. You can see a bulging into the abdomen because the intestine have no space, no enough space for its big size in this stage, and thus some of the intestinal parts protrude into the proximal parts of the umbilical cord. In the third month, the fetus, it is not an embryo by now, having a human-like face because the eyes becomes anterior or ventral, still the ears are lateral, and the limbs become more proportional to the size of the body. You can see that the size of the head is about half of the crown rump length. You can see also that this is the amnion and that it fills the uterine cavity in that stage. This is the chorion frondosome attached to decidua basalis. This is the chorion leaf with the decidua capsularis. This is the decidua parietalis. So you have decidua basalis, chorion frondosome, decidua capsularis, chorion leaf, and decidua parietalis. Next, to the third week is the 5 to 6 months. Fetus, in which you can see that the head is about 1/3 of the crown rump length, the limbs are more elongated, and that the embryo shows at the fifth month some hair, which is not well represented in this plastic sample, which is called lanugo hair, and so on. In the 6th month there will be wrinkling, and so on, as you will reach to the features of the full-term baby, which is normally developing and is demonstrated in that figure. Everybody could appreciate that this is the full-term baby, right?